Good afternoon, and welcome to What's Lecanemab and What's Next. This is a community conversation with the co-directors of the Penn Memory Center, David Wolk and Jason Karlawish. We're going to talk a little bit about sort of the history of lecanemab, what brought us to the FDA approval of the drug, and what we're looking at for this year and beyond for Penn Memory Center patients, research participants, and the community around us. So without further delay, I will turn it over to Jason Karlawish and David Wolk.
Good afternoon. Hi, I'm Jason Karlawish. I'm a co-director of the Penn Memory Center and associate director of the Alzheimer's Disease Research Center. And it's a pleasure to be here together with my friend and colleague, David Wolk.
Hi, everyone. Thank you for attending. I am also co-director of the Memory Center and director of the Penn Alzheimer's Disease Research Center.
Let me walk you through the run of our time together today. But before we do that, just some disclosures. I'm a site investigator for clinical trials sponsored by Biogen and Lilly, and I serve on the board of advisors for eFamily Care. Dave?
I am a paid consultant to Eli Lilly, GE Healthcare, and Kynaps. I'm on a data safety monitoring board for functional neuromodulation, and I've been a site investigator for a clinical trial sponsored by Biogen.
Here's the agenda for what we'll talk about today. I'll begin by telling the story so far of how lecanemab became Leqembi. Next, David will take over and explain the details about what is lecanemab. Then David and I will walk through several use and non-use and some tough in-between cases for prescriptions for the drug. And then finally, we'll tell you about what's next at the Penn Memory Center. This talk is being recorded and archived, so you can come back and watch it at a future date as well. So let's start out. The story of how lecanemab became Leqembi. This may seem a little what and why, but I think it's important to know where a drug came from, because where this drug is at right now is in a rather unusual space, and it affects your ability to have access to it and my ability to prescribe it. So let me explain what's going on. Not too long ago, earlier this month, Eisai incorporated its application to the FDA for their drug lecanemab was approved by the FDA, and so lecanemab became Leqembi, which is its trade name now for the company to market the drug. It's important to understand, though, that the FDA's approval was not the FDA approval that we typically think about when FDA approves a drug. It was an approval using a very unique regulatory mechanism called accelerated or conditional approval. Accelerated approval allows a drug to be approved so that a company can market it for sale, based not on the showing that the drug is safe and effective for the treatment of a disease, but instead based on evidence that the drug affects a marker of the disease, and the effect of that marker is reasonably likely, but not yet proven, to translate into improving a patient's health and well-being. Hence the idea of it being conditional approval. The regulations that were developed to allow this came from the 1990s, when there was concern that the pace of drug development and the evidentiary standards used to approve drugs for HIV, a serious and untreatable illness, were inappropriate. Alzheimer's, of course, is a serious and untreatable illness. Hence, these regulations were used for the approval of lecanemab. So I've mentioned this already. Let me drill down a bit more. As you know, typically, when FDA approves a drug, they're saying that there's substantial evidence that the drug is safe and effective. That's regular approval, and that's most drugs. The vast majority are approved under that mechanism. In rare exceptions, for serious and life-threatening illnesses for which there's no effective therapy, accelerated approval can be used. And the standard there is substantial evidence that the drug has an effect on a measure of the disease, and that that effect is reasonably likely, but not yet proven, to translate into clinical benefit. When a drug is approved under accelerated approval, that approval is conditional upon verifying clinical benefit in a subsequent confirmatory clinical trial. And FDA has, although not often, pulled drugs that were approved under accelerated approval because of the failure to either conduct the study to confirm benefit, or the failure of subsequent studies to in fact confirm the drug is beneficial. This has been in the case for drugs for various cancers. In the case of Leqembi, the marker that allowed them to make this judgment was a reduction in beta amyloid measured using a PET scan. And later, when David Wolk talks about the drug, he'll explain this measure of beta amyloid using a PET scan. It's a key measure to decide whether someone is eligible to take the drug or not eligible. When FDA reviews a drug, it sort of has gone through at least 2, if not 3 phases of development. The phases of FDA review begin with phase 1, showing that a drug is basically safe and some hint of how to dose it. Phase 2 to further confirm safety and better verify how to dose it. And then it's phase 3. And in those phase 3 studies, the goal is to show that the drug improves, helps, otherwise is beneficial to the well-being of patients, and of course is safe. Lecanemab, was approved on the basis of a phase 2 study, on the basis of an early phase study designed to show safety and dosing. It was not approved on the basis of a phase 3 study. And this is where things get a little funky, and you might be wondering, I kind of get why he's telling me all of this. Well, remember, the logic of why a company would want accelerated approval is because the moment you have approval, whatever that may be, you can market a drug. And indeed, this is from Lecanemab.com. You can visit the website. And this is Eisai's, the company's website, promoting their drug, allowing you to sign up for alerts about the drug, etc. So already the marketing has begun. That's what drug companies do. They produce a product they believe in and value, and they sell it. So that's the value of accelerated approval. Well, a little bit more about how Lecanemab became Leqembi. I told you it was approved under accelerated approval on the 6th of January. But what's weird is you probably heard about last year, towards the end of the year, just before Thanksgiving, around that time, that Eisai announced the results of a phase 3 study that showed that Lecanemab was beneficial for persons with Alzheimer's disease. That study was called Clarity. You probably heard that later in the month of November, Clarity's results were presented at an international conference and published in the New England Journal of Medicine, and that the results showed on measures of cognition, function, quality of life, caregiver burden, that the drug performed better than placebo. And David Wolk will go over all of these points in much greater detail in just a few minutes. And so what's gone on here is that the company used the phase 2 trial results done a while ago to get accelerated approval. The phase 3 wrapped up not too long ago, the announcements in the fall, but those data have not been submitted to the FDA yet for review and a decision for full approval. So right now we have a drug that's available under accelerated approval, not full approval, but we have a clinical trial that would be used for full approval, whose results you can go and download off the internet from the New England Journal of Medicine's website or watch the presentation at CTAD. Where did this all come from, and why are we in this sort of odd situation? Well, some of it goes back to another drug's history, which I'll tell you about in a minute. But just a last bit on Lecanemab is the price for the drug has been set at an annual price of about $26,500. And the Institute for Clinical and Economic Review reviewed the drug's performance in clinical trials and calculated a cost-effective annual price should be between about $8,500 and $20,000. So that's a perspective on the drug's value to society, and I think some of the emerging issues that will surround what's it worth and how much should be paid for it. But let's go back and wrap up here on the history of it. Lecanemab is very much a product of the history of another drug that many of you know about, a drug called aducanumab, made by the company Biogen. It was approved under accelerated approval about 2 years ago in June. Its story is a little odd. I won't dwell on it. You can go to our website, search under aducanumab and learn more about it. But there they had a phase 3 trial, which was incomplete and of mixed results, and that was used to give accelerated approval. This caused an enormous controversy over the quality of the evidence and FDA's decision. I think a justified controversy. Medicare, CMMS, looked over those results and decided that they did not feel that aducanumab had evidence that supported that it was reasonable and necessary for patient care. And so Medicare declined to cover the drug, declined to pay for a prescription. Most other Medicare insurers, indeed all the Advantage plans, also made similar decisions, as did the United States Veterans Administration. Medicare also issued guidelines that said we will pay for this drug if you're in a study designed to prove that it in fact treats a patient. And so the drug sort of died on the vine, needing more study and no coverage, if you will. It was those regulations, though, and the precedent of aducanumab that allowed Eisai to apply for this accelerated approval for their drug. But now, of course, we have a phase 3 study, which still needs FDA review and hopefully for full approval. So that's where we're at right now with this drug. We're actually awaiting Medicare's decision about whether they'll pay for it. I have a hunch Medicare is simply waiting for the full approval to then make a decision. One last bit of piece of news, just to sort of tie all these drugs together. And I've heard about this drug lecanemab. What about that other drug that Biogen made? Well, there's a third drug that's been in the news, and that's a drug called donanemab, and that's owned by and being studied by the company Lilly. And you may have heard, not just last week on January 19th, that FDA declined to approve Lilly's application for accelerated approval. They granted approval for aducanumab, they granted approval for lecanemab, but not donanemab. Why? FDA's explanation was very precise and to the point. FDA said that for accelerated approval, we need at least 100 people who received the drug for at least 12 months. And the data that Lilly presented did not include data from 100 patients who met that criteria. And so the application was tabled, was rejected. Myself and most of my colleagues are of a similar view. Not terribly worried about that. What we really want to hear about are the results of the phase 3 trial that Lilly has done or is doing, soon to wrap up, of donanemab. This spring, in about 3 months, we hope to hear the results of that study. And those will be the results that we want to take a look at, because those are the results that will inform whether this drug should be part of clinical practice. So that's the history of how lecanemab became Leqembi. It's complicated, it's odd, but I think you need to know it because it reflects some transformations in what's happening at FDA. And I think it helps explain why am I hearing about all these drugs, some of which are very controversial, aducanumab, others are not so controversial, Leqembi. And now I'd like to turn it over to Dave to give you more details about Leqembi. Dave?
Here we go. So I think that was a really nice history of what's gone on with lecanemab and or Leqembi. Now I want to tell you a little bit about what the drug is and what's the data that we have that support that this drug may have an impact on the clinical lives of patients living with Alzheimer's disease. So lecanemab is an antibody. It's an antibody like the antibodies that everyone produces in their body themselves, except in this case, it's an antibody that we give to patients through an infusion. And the antibody targets amyloid in the brain. And as many of you know, one of the sort of hallmarks of Alzheimer's disease are the so-called amyloid plaques of the disease, which are when amyloid proteins clump up into a insoluble form and create these plaques. Well, there are other forms of amyloid before it kind of turns into those plaques that are more soluble and smaller in terms of the number of molecules, and those are called oligomers and protofibrils. And the reason why that's potentially important is that this drug particularly targets them. And those forms of amyloid, at least in some studies, have been suggested to be the most toxic form of amyloid in the brain. That being said, the antibody actually binds to also the plaques as well to some extent, which might reflect some of its mechanism of action as well. All right, having troubles moving ahead my slides. I think I figured it out. So how did the dosing for lecanemab, how was that decided? And as Jason mentioned earlier, there was an earlier phase 2 study of lecanemab where the goal of that study was really to find out what's the sort of optimal dose of the medication. And so they looked at a variety of different doses of the medication and determined which doses best hit the target of reducing amyloid in the brain as measured by amyloid PET, as well as to some extent, to what degree did these different doses affect clinical outcomes? Although, again, the primary goal of this study was more around dosing. And what was found was that the highest dose that they used which required a twice weekly infusion or an every 2-week infusion of the drug, so a drug given at 2-week intervals, in green here, showed the greatest reduction in a measure of amyloid in the brain relative to other doses of the medicine. And in fact, there was some data, although it was a relatively small study, showing clinical benefits associated with this higher dose of the drug. The one thing I will point out is that the size of these effects from this earlier phase 2 study are actually very similar to the phase 3 study I'm going to show you in a second. And I think that provides some convergence of data to support that what we're seeing in the phase 3 study isn't some kind of fluke. So with that data in hand, as Jason mentioned, the company went forward with a study called Clarity AD, which was the phase 3 sort of definitive study to see if lecanemab works. And they were using the dosing described in the prior slide. And they enrolled about 1,800 individuals, so a large study in patients with mild cognitive impairment. Those are people who have memory and thinking issues that are beyond what we expect for age, but not enough to really impact their function in a significant way, or patients with mild dementia where function is affected to some extent. People need a little bit more assistance in their day-to-day activities. And we'll talk a little bit about the meaning of those terms in a bit. The other sort of key point is that everyone in the study needed to have an amyloid PET scan, and it had to be positive. It had to show evidence that they had underlying pathology of Alzheimer's disease. And what they looked at over 18 months of the trial, so if you look here on the x-axis here, these are the number of months. So 18 months was the end of the placebo-controlled study. And the y-axis here is amyloid PET measures. And so the only thing that's important to know here is that the higher up you go from 0 means you're gaining amyloid over time. And the placebo group showed a little bit of elevation in amyloid over time, which is typical of patients with clinical symptoms of Alzheimer's disease. There's a little bit of an elevation of amyloid, but not a ton after the amyloid has already risen to a positive level. But what was seen in the lecanemab group was a really dramatic reduction in amyloid in the brain. And in fact, by the end of the study at 18 months, 2/3 of the patients no longer would be considered amyloid positive, which is really a remarkable outcome that one, can even see the pathology of Alzheimer's disease in the brain, and 2, you can give a drug that can actually remove that from the brain. The problem is amyloid in general is not very strongly associated with the symptoms of Alzheimer's disease. It may be a necessary step to get symptoms, but it's not the thing that's really linked to injury in the brain, loss of synapses or the connections between cells in the brain that are important for thinking. And so if I wanted to feel confident that a drug that targets amyloid might have an effect on thinking, I would want to see that it had an effect on downstream changes that occur in the brain that are more linked to thinking. And in particular, the other main sort of thing we see in Alzheimer's disease are the tau tangles. So there are plaques, and then there are tangles, and the tangles are made up of a protein called tau. And they very strongly associate with the memory and thinking problems of Alzheimer's disease. And I think really compellingly, in the lecanemab study, using 3 different measures of tau, one in plasma, one in cerebrospinal fluid, and one with amyloid PET imaging, in all 3 of these measures, there was evidence that the lecanemab group had a relative reduction in tau or less of a rise on tau PET than patients in the placebo group. And so it really strongly hit that downstream target as well. And that, to me, was actually something that was quite compelling, that maybe this drug would have a clinical benefit. There were some other measures that they looked at of other kinds of downstream things that tell us that this drug might have an effect on more than just amyloid. Another was a measure called neurogranin, which is a marker of synapse integrity. And what was seen here is in the placebo group, there was no change to a rise in that over time, whereas in the lecanemab group, there was a reduction in this marker, suggesting better synaptic health with lecanemab. And again, synapses are important for the connections between cells in the brain that help with thinking. Another measure called GFAP is a measure of inflammation in the brain that might have negative effects on the progression of disease. And again, the lecanemab group showed a reduction relative to the placebo group. So all this was really promising, but of course, the thing you really want to see is, does this affect people's thinking and function? And there was also pretty consistent data across all of the measures that were looked at in this study that favored the placebo group, or favored the lecanemab group versus the placebo in slowing down, not halting, but slowing down the progression of disease. And in fact, there were 5 different measures of cognition and function that were looked at, and all 5 of them very significantly showed an effect of lecanemab over placebo. The one that you probably have heard the most about is something called the Clinical Dementia Rating Scale, Sum of Boxes, which was sort of the primary outcome measure that was looked at in this study. This measure is a measure that's based on a kind of structured interview between a physician or a clinician and a patient and that patient's care partner. And based on that, one can assess thinking and function in that patient. And what was seen was that, again, lecanemab didn't stop the progression of disease, but it slowed it down relative to the placebo group by about 27% at the end of the 18 months of the study. Maybe even more compellingly, there was another measure that measures activities of daily living, which is essentially your ability to do day-to-day functions like the checkbook, et cetera. And what was seen here, again, was that there was a slowing in the lecanemab group relative to placebo, and here a 37% difference. One of the things that was also noted in all of these different measures is that there seemed to be a widening of these differences over time by 18 months. And so I think one thing we don't know yet is whether or not this widening continues to occur after 18 months. Maybe it stops and you don't get any more widening. And so the effect that you get after 18 months is the effect you get moving forward, or maybe, the two groups come closer together. We don't know. And that's why it's really going to be important to get data from some of the open label phase parts of this study. So people in the study were allowed to continue on after the 18 months of the study. But also, as we move this into clinical practice, I think we're going to really need to follow people over time and really quantify how they do to better determine, are these effects even greater than what we see at 18 months, or do they kind of peter out over time? One other way of thinking about a drug that could slow down Alzheimer's disease is to look at its ability to slow down or delay a patient reaching a particular clinical point. And so another analysis that was done in this study was to look for what proportion of people went on to a higher score on an overall measure, something called the Global Clinical Dementia Rating Scale. And what was seen at 18 months of the study was that in the placebo group, about 32% of the people or so went to that higher, that more impaired level of function and thinking by the end of the study. Whereas in the lecanemab group, about 24% went on to that level of progression by the end of the study. So about 25% less people went on to show this level of progression. Again, a modest effect, but an effect that's there. The other, I think, really important point here is that 68% of people over 18 months in the placebo group didn't go on to progress to this higher level of Alzheimer's disease. And I think this sort of emphasizes the point that this is, even though it might not feel this way, at least with the way we measure it, it's a slow-moving disease. And over 18 months, there's not a lot of change, even if you're untreated. And so trying to show a slowing of a slow-moving disease has been really one of the big challenges in the field, and makes studies like this lecanemab study, a challenge. And I think impressive that any effect can be seen in this context. So I think the big question you're all probably asking is, okay, you're showing me these graphs and figures, and it shows that that lecanemab slows down the disease, but is this effect, am I going to notice this? Does this effect have any real meaning for me? And it is true that this is a modest slowing of the disease, but it certainly doesn't halt the progression of the disease. The disease still progresses even when taking this drug. The measure on the clinical dementia rating scale, sum of boxes, is about 0.5 points. It's an 18-point scale, so maybe that's not such a big change. However, one should keep in mind that in this study, these were very, very mild patients. And so we were really using just the low end of that scale. And the mean sum of box score of the entire group was 3.2 at baseline. And even in the placebo group, there was only a 1.7 point change over the 18 months. And so we're talking a very small window for which that 0.5 change or benefit from the drug can be seen. And I think one important thing is, how do we measure these things? When you look at the scale itself, 0.5 points based on how we score can be the difference between something like slight benign forgetfulness versus more moderate memory loss that interferes with day-to-day, with everyday functions. And so that kind of change can have an impact in patients' lives. Maybe even more easy to see the potential clinical impact is using that activities of daily living scale that I mentioned earlier, where the lecanemab group did 2 points different, or 2 points better. And again, looking at that scale, a single point, particularly at mild phases of the disease, can reflect a difference between managing one's own finances independently versus with assistance. And so a couple points on that scale might make a difference in someone's day-to-day life in a real tangible way. Another way to think about the clinical impact of this drug is to think about measures of quality of life or burden on both the patient as well as on the care partner. And so they actually looked at this in the study using several different scales. And actually all of the scales favored the lecanemab group. So patients rated a higher quality of life, as did their care partners. Care partners felt they had less burden on the drug than off the drug. And on some of these ratings, the effect was somewhere on the order of 50%, ranging from about 23% here to, I think, 56%. So it's great to talk about benefits, but anytime you talk about benefits of a drug, you have to talk about what are the potential side effects of the drug. And probably the one that most of you heard about is the so-called swelling and bleeding that can occur with this disease or with this medicine. And those have been referred to in a technical sense as amyloid-related imaging abnormalities. There's amyloid-related imaging abnormalities, edema, which reflects some leakage of fluid out of blood vessels. That's that swelling that people refer to. And then there's amyloid-related imaging abnormalities, hemorrhage. And that's when blood can leak out of blood vessels. That's that bleeding. Although the bleeding more correctly is, and almost always, is something we call microhemorrhages, which are really tiny little bleeds in the brain. What we can see here on two of the images are that edema. So this bright area on this scan is an area that we would call ARIA E, because there's a little bit of an increased signal here, which reflects fluid. We can see that also on this patient here. These are patients that were treated with a drug like lecanemab, although they weren't actually treated with lecanemab. In the middle figure, or the middle image, we can see the microhemorrhages that I was talking about, which are these little dark circles. One really important thing to know here is that this is a patient who wasn't treated with a drug like lecanemab, because these little microhemorrhages are actually common in Alzheimer's disease, because Alzheimer's disease is associated with amyloid that gets in blood vessels called amyloid angiopathy, causing blood vessels to be leaky themselves. And so some of this bleeding that we see is actually really intrinsic to Alzheimer's disease. It's just that lecanemab increases the risk of that. So how much does it increase the risk of these? About 2 to 1. So in the lecanemab group, about 21% of patients had this so-called amyloid-related imaging abnormality, either the edema or microhemorrhages. whereas about 10% of the placebo group had it. Again, 10% of the placebo group still had this side effect because it's part of what we see with Alzheimer's disease, particularly the microhemorrhages. However, almost 80% of these changes were asymptomatic. And that's part of why originally these were called amyloid-related imaging abnormalities, is because they were picked up with what we call surveillance MRIs that are obtained even though a patient has no symptoms, but just to assess whether they're there. And the vast majority of them were found that way. That being said, about 3.5% of people in the lecanemab group did have symptomatic edema and microhemorrhages, whereas only about 0.2% of the placebo group did. Again, though, these symptoms tended to be very manageable and very mild, usually including headache, visual disturbance, or confusion that would return to normal after the drug was held for a period of weeks to months. And many people could go back on the drug after it had been held for a period of time. There were serious adverse events, and in clinical trials, that's usually defined as when someone needed to go to the hospital for something, or when an event was considered quote unquote life-threatening. And there was more risk in the lecanemab group than the placebo group, but the risks were pretty close, actually. It was 14% versus 11%. So just a few percent difference. One thing to note, that 11% of serious adverse events in placebo is a reflection of the fact that that Alzheimer's is a significant illness, and these are older patients, and they have many other kinds of medical problems that can lead to serious adverse events. Death was no different in lecanemab versus placebo in the trial. So the death rates were the same, and it was a very, very small percent of patients. However, in the open label extension trial that was done after the 18 months of the study, there actually have been 3 reported deaths that have been, I think, pretty well publicized in the media, 2 of which were associated with blood thinners, and 1 of which was associated with a so-called clot buster, or TPA, in the setting of stroke. And I have to say, even in patients with Alzheimer's who have some of these microhemorrhages and aren't on lecanemab, I have concerns, or it gives me pause to treat with those medicines. In the context of being treated with lecanemab, I get even more worried. And so I think many of us in this field feel like this might be something that would be a contraindication to wanting to start the drug. One other just really important point related to the side effects of the drug is that one of the main genetic risk factors for Alzheimer's disease, called apolipoprotein E, the E4 allele, of which each of us, everyone has 2 copies of apolipoprotein E, and the E4 version of it increases your risk of Alzheimer's by about 2 to 3-fold. It's the most common, again, genetic risk factor for Alzheimer's. And what was seen in the study is if you had 1 copy of that E4 allele, you had about a 2 times risk of the edema and kind of a slightly higher risk of the microhemorrhage. So a little bit of increased risk, but not tremendously so. However, if you carried 2 E4 alleles, the risk goes really way up to 6 times the risk of the edema, and almost 4-fold increased risk of the microhemorrhage. And this has actually been seen with other drugs similar to lecanemab as well, that being a carrier of both, having both E4 copies makes you a very high risk. And so I think when thinking about risk benefit, apolipoprotein E may be an important piece of us making decisions about whether or not we want to consider the drug, and whether or not families and patients want to take on those risks, because it's likely the case if you're, for example, an E3 individual, so not a carrier of the E4 gene, that you have relatively low risk and potentially higher benefit. Whereas if you're in the E4 carrier, your risk may be higher, but your benefit also may be lower. And so these are decisions and discussions that I think we'll all need to have with patients and families. And with that, we're going to move on to talk about some of the sort of decision-making around cases of individuals who we might consider lecanemab. and I'm going to turn it over to my colleague to talk a little bit about how to think about these cases.
Thanks, Dave. So some guidance in thinking about these cases. Remember, these are cases about what we would, when I say use, it would mean that a clinician like myself or Dave would say, I will write a prescription. In contrast, non-use would be, I'm not going to write a prescription. And the in-between are, this is a really tough call. When you think about these cases, a couple points to keep in mind. Don't overread one detail. These cases are intended to illustrate the clinical points that we're thinking about, the top line ones, not actual individual patients. Another key point is, across all the cases, you'll see us speaking about someone's daily activities. Daily activities describes the kind of things that we do to live our adult lives. For example, using transportation to get from one place to the other, whether that's the bus, Uber, your bicycle, or your feet and your iPhone. Shopping, cooking, cleaning the house, taking your medications, having bills come in, paying them on time, balancing the necessary accounts, sorting out the financial paperwork, using technology to communicate, computer, etc. Collectively, these are the kinds of activities that are bundled together into a category that you may have heard of called the instrumental activities of daily living, to be distinguished from what are often called the basic activities of daily living, which describe bathing, dressing, grooming, feeding, and toileting. The reason why I'm dwelling on these activities of daily living, these IADLs, is they're really key to define someone who has cognitive impairment who then might be a candidate for this drug, because they need to have either inefficiencies or impairments, disabilities in the daily activities. So let me develop those points a little bit more. When we talk about inefficiencies in daily activities, what we're describing is someone who says, I'm still doing the finances, but gosh, it takes me so much longer. Every now and then I'm making a mistake, I catch it, etc. I'm still doing the cooking, but I've been making some mistakes, or it's getting a little simpler, etc. The description of someone who has largely inefficiencies in daily activities is a classic description of someone with what we call mild cognitive impairment. They have cognitive impairment that's detectable on testing. They feel it, others around them notice it, and it's causing them to take longer to do those daily activities, at least some of them. In contrast, disabilities means mistakes are happening, there's enough trouble doing it that if someone else doesn't help out, or something, some device that wasn't used before, needed before, there will be problems. Errors in the checkbook, troubles getting a meal prepared, getting lost while driving. We could run that list. I'm sure for those in the audience, there's some very vivid stories that you have. When we hear about disabilities being present, that's when we begin to start thinking about mild stage dementia. So those are two key concepts to think about. Again, don't overread a detail. These cases illustrate clinical points, clinical details that we'll be thinking about as we think about whether this drug is a drug to be prescribed for a patient or not. What we don't present in these cases, and it's something I think many of you are thinking about right now, are the experiential details of what it's like to be a person with mild cognitive impairment or mild stage dementia, what it's like to experience that disease. In particular, what are the patients' and families' intentions? What are their desires and beliefs as they learn about this drug? Intentions such as the belief that the drug is beneficial, that this drug, now that I've heard what Dr. Wolk has said on balance, I think is worth taking, or that this drug is too risky. Now that I've heard what you've told me about it, I don't think this is a drug for me. Another belief and intention that people may have is, where I am right now with this disease, I want to kind of stay here. I want to extend that period as long as possible. Or I don't. This has gotten too bad already. I don't want to extend that period. Those are deeply personal issues, highly ethically charged. We're certainly here to help people think through them, but they're not the kind of things we're going to talk about in these cases. And so with that, let me turn it back to Dave to walk through the cases.
Okay, great. So I'm going to start off with the first case, and I'll ask Jason to step in when he has thoughts or additions he wants to add. You'll see a pattern to these cases in terms of the description, as we're going to change pieces of them to show how those different pieces impact how we might think about whether or not to consider the drug. So the first case is a 76-year-old who has declines in memory, executive functioning, which is your ability to do things like multitasking or to solve complex problems. Patient also had a little bit of decline in word finding as well. And this patient had inefficiencies in their daily activities. So as Jason said, these would be individuals who were just slowed down, but able to do many of the day-to-day activities that they could always do in the past. And this is an individual we would say has mild cognitive impairment. Their MRI showed some degree of brain atrophy, which we often see in mild cognitive impairment. So a little bit of shrinkage in parts of the brain, usually in parts of the brain involved in memory, but didn't show any evidence of those microhemorrhages I mentioned earlier. They went on to get an amyloid PET scan, which was elevated. So it was positive for having amyloid in the brain. And so at this point, one would say their mild cognitive impairment was likely due to Alzheimer's disease. And they were an E3 carrier. And so in many ways, this is kind of the ideal case for lecanemab, as this is a patient who fits within the kind of clinical range that the study was done. It's someone who doesn't have a lot of other medical issues, doesn't have microhemorrhages suggestive of amyloid angiopathy that we can at least see on the scan, and is a non-carrier of the E4 allele, so is at relatively low risk for some of the side effects of the medicine. And so this would be someone we would have a conversation about whether or not they were interested in trying the drug. The next case, we're changing it up a little bit by saying that this patient that we're seeing here is similar to the other one, but now instead of just inefficiencies in some of their activities, they're requiring assistance with them. So maybe they require assistance managing their medicines, managing the checkbook or cooking, et cetera. And so this is someone who has mild stage dementia. MRI could look pretty similar to the last patient. And actually, we often don't see too much of a difference when people have mild dementia versus mild cognitive impairment on MRI. This patient also had elevated amyloid, so we think their mild dementia is due to Alzheimer's disease. And they also are an E3 carrier. And so again, like the last case, they fit that clinical window that was seen in the clinical trial, or that was a criteria to be in the clinical trial, and are at relatively lower risk of some of the side effects of the drug, being a non-carrier of E4.
I think a point that you're beginning to maybe see here is these bullets are in a sort of an order, because one of the things we're thinking about as we're working with our colleagues here at Penn Medicine is kind of the steps that you would follow after you learn about this drug, like learn about it at something like watching this video over and over again in our materials on our website, is first that careful history, physical and exam, the two first bullet points. Then if things look okay, an MRI, if that looks okay, then testing for amyloid. And if that passes for showing elevated amyloid, then ApoE testing. Some of the cases will vary on these points, but that's kind of the way we're thinking about the steps one would go through moving along to receive this drug. Dave, carry on.
So the next case here, again, some of the same areas of thinking are affected, but at this point, most of this person's activities or instrumental activities of daily living require assistance, that we would say there's a disability, that they can't do these on their own anymore. And so this is what we would classify as a moderate stage of dementia, and is someone who was further along than what was included in the lecanemab study in terms of criteria to get in that study. Other factors for this patient are exactly the same. The MRI, might show a little bit more atrophy by this stage, but no microhemorrhages. The amyloid PET confirms that Alzheimer's is the cause, and the E3 status is the same as the prior cases. And so one might say, well, in the trial, yeah, they didn't include moderate stage dementia, but I still want to try this drug because, I want to slow down the disease. And so why shouldn't I? And, lots of drugs are used off-label from what they were originally studied as. Well, I think in this case, part of why we would be hesitant to try the drug at this point is that there's evidence about two things. One is that it might be the case that individuals who are at a later stage of disease would be even more likely of those side effects that we talked about, because as the disease progresses, people tend to have more of that amyloid angiopathy, which can cause that edema and bleeding that we spoke about. And there's some reason to believe, based on the science, that treating at a later stage of the disease is less likely to slow it down. And so, I think most of us feel, without real strong evidence to the contrary, that the benefits are going to be smaller in this population while the risks are going to be higher. And therefore, we would tend to not want to use this drug in this instance.
Now, a clever viewer may say, Karlawish, in the last slide, you said these are bullet points because they represent the stage of your evaluation. Why is it that you, after discovering the person had moderate stage dementia, still went ahead and got the MRI, the amyloid scan, and even an ApoE result? And so you're correct. You've caught me. It's not an error. Pretend that this person had prior MRIs for diagnostic workups and perhaps was in a research study or other that got an amyloid PET scan and also an ApoE result. And certainly for a diagnostic workup, we would be getting things like an MRI and maybe someday also amyloid or other kinds of studies for biomarkers. But I do want to emphasize that once it's sort of established the severity of disease is either not mild cognitive impairment, but just say subjective impairment or moderate stage dementia, the decisions around whether lecanemab is going to be prescribed would be made at that point, and no further need for additional testing would be done. Dave?
Yep, that's a great point. So the next case, again, same sort of set of cognitive symptoms or thinking symptoms. This patient has inefficiencies in some of their activities of daily living. So again, would be considered mild cognitive impairment. The MRI might look pretty similar to the last patient, but this patient went on to get an amyloid PET scan and it was negative. There was no evidence that there was amyloid in the brain. And for obvious reasons, this would be an inappropriate case to use the drug, because the drug is targeting the amyloid of Alzheimer's disease. And so if you don't have that amyloid, the drug's not going to be effective. Here's a bit more challenging of a case. This is, again, very similar to the prior ones. This patient falls in that mild cognitive impairment category, so that falls within the prior studies inclusion criteria. The MRI looks similar. There's no microhemorrhages, so not a lot of evidence of amyloid angiopathy. And the PET scan is consistent with Alzheimer's with elevated amyloid, but they're an E4 carrier. And so again, as I mentioned earlier, these individuals would be at the highest risk of the side effects of the drug. And that may give pause as to whether or not, relative to the very modest benefits of it, one would want to proceed with taking the drug. And certainly it would be something, as a clinician, I would have some concerns about, and would definitely want to have a frank conversation with families about their expectations and how they weigh risks and benefits. But this would be a much higher risk person.
The other part of that conversation with the family, of course, would be that having established that the individual has 2 copies of the ApoE4 gene, it means that any of that individual's blood relatives, children in particular, children who are blood relatives, meaning they're the biological parent, that child has at least 1 copy of the ApoE4 gene. These kind of issues will be explained more when we develop materials and have them available around thinking through the ApoE gene test. But the key point here is an ApoE gene result is a result in an individual, but it's also a result in a family, because of course genes are passed on through inheritance.
And I'll just add, we have genetic counseling experts here at Penn, and that may be a resource that one could consider when we're thinking about doing this testing as well. All right, this case, again, a lot of the same features, mild cognitive impairment, level of activities of daily living. MRI looks the same, no microhemorrhages. But this patient has other comorbidities, other medical issues that increase their risk of vascular disease. They have hard to control blood pressure, and they have heart failure, which can sometimes lead to funny heart rhythms that can result in people needing to be on blood thinners. This patient has an elevated amyloid, and they're an E4 carrier, but they have only 1 of the E4 genes, so maybe at a little bit more risk but not, the full risk of someone who carries both of those E4 alleles. And this would be a case that would require a conversation and thinking a little bit about what the risks and benefits are, because there's the risk of the drug in this individual, but there's also the risk that these medical issues may lead to treatments that would be incompatible with the drug, or at least things that would be concerning to give with these patients. So anticoagulation, so think medicines that thin the blood, or as was in that one case that I mentioned earlier, a clot buster in the case of an acute stroke that someone would have, that could be very risky in the context of taking lecanemab. So again, these would be considerations and things that we would have to think about in someone like this. And I think it would be a tough call for those reasons.
And just simply to kind of expand our kind of thinking in this conversation, and this is the beginning of a conversation, there's somewhat of a cruel irony to these drugs. They're designed and they're working to help a brain remain healthy, to help a mind remain healthy. But the very side effect that they can cause is damage to that very same organ, the brain. And I recognize I haven't helped you think through that perhaps, but I think I've helped frame what is the disquiet that you might be feeling. I've often thought myself, if this side effect was, say, to the liver or the kidney or whatnot, it'd be kind of a different kind of set of calculations, or not calculations, but ethical considerations that one would be engaging in. Dave.
This is another case with mild cognitive impairment, but this is someone who's already on a blood thinner for one of the heart rhythms that requires it, called atrial fibrillation. And so this is someone that I think many of us would feel very uncomfortable about starting a drug that increases your risk of bleeding, given that the blood thinner the person's on would further increase that risk as well. And then
indeed, it was the case in the trial, by the way, that people could be on blood thinners, and what we saw was greater rates of bleeding, et cetera, in those individuals.
Finally, this is the last case. Similar case. This case is a mild dementia case, but we know from the start that this is someone who came to see us who had a pacemaker in place and can't get MRI scans. And so you might wonder why, if you can't get MRI scans, would we sort of stop things there and say, well, we can't, consider lecanemab in you? And that's because, as I mentioned earlier, the edema, the fluid, and the microhemorrhages that we see with this drug most of the time are detected in people who are asymptomatic, with MRI scans that are done periodically to assess for safety. And actually, if you see these changes, you halt the drug for a period of time until they resolve, and then can re-engage with the drug again in the future. If we can't measure what's going on in the brain, if you have a pacemaker or some other contraindication to getting an MRI, it's really not safe to take this drug in that context. And so that's why this case would be considered an inappropriate use of lecanemab.
Early on in the course of, if one chooses to go ahead taking the drug, infusion every 2 weeks are frequent MRIs before, and then in the months to follow, frequent MRIs. So just to reiterate Dave's point, someone who can't tolerate or otherwise get an MRI is not going to be a candidate for a prescription for this drug. One more, Dave.
Oh, there is one more.
There's always room for one.
So this one, similar to some of the others, mild stage dementia, elevated amyloid on a PET scan, and this MRI showed 4 microhemorrhages. And when people have microhemorrhages, even without treatment on lecanemab, it provides pretty strong support that they have some degree of amyloid angiopathy in the setting of Alzheimer's disease. And so this is also someone that we would have concerns. I certainly would myself, and I suspect Jason agrees with me, would be someone that we would think would be at higher risk for bleeding in the future, because, and also the edema, because of the fact that they already have relatively obvious and manifest amyloid angiopathy. And again, it would at least merit a strong conversation about the potential for an increased risk in this individual.
Exactly. And indeed, again, that MRI would have been done before even the PET scan. We wanted to give you examples of people. For whatever reason, I had a PET scan done as part of research or another clinical trial that I was in, et cetera. But once we see those microhemorrhages, that raises real concern about causing more microhemorrhages, not because of the amyloid angiopathy, which is what this person has, but because of the drug. So what's next at the Penn Memory Center, Dave? Well, as I explained to you in my beginning is my end, to paraphrase from T.S. Eliot, this drug was approved under accelerated or conditional approval simply on its ability to lower beta amyloid as measured by PET scan. I think, frankly, a very controversial decision by FDA, and one that still causes a lot of dissent and controversy within the field. Having said that, we know there's a phase 3 study that's done. I read the paper. David presented to you slides that were taken from that paper. It's a very well done study, very well done paper. So we're waiting for FDA to review those data and decide on full approval. At that point, we think CMS and Medicare will really have the kind of evidence they need to make a decision about whether this drug is reasonable and necessary for the treatment of patients, and therefore is a drug that they'll cover. Meanwhile, at the Penn Memory Center, we're working to create a pathway that will move a patient and family from their interest in and inquiries about the drug to a decision to take it or not to take the drug. This will feature patient and family educational materials that you'll ponder on your own time, and a step-by-step process to determine if the drug is a fit for you, is right for you. And in some sense, that education has begun now and will continue now as you have the chance to come back and watch this video over and over again, because it is just so fascinating and beats any other. No, anyway, and also on our website, we'll have a variety of educational materials as well. Because, you know, when you enroll in a math class, the first class is not the last class to learn algebra or geometry or calculus. It's the first of several classes to finally say, I now understand algebra, geometry, whatever it may be, or English. Pick your discipline. There's a lot of new information here about how to think about Alzheimer's, more nuanced thinking about the staging, the use of biomarkers, genetic testing, the risks of bleeding and hemorrhage. A lot of information. We don't expect to have been able to digest it all and figure it out now, but this is available for you now to come back and look at along with other items on our webpage. And then finally, we're very keen to participate in a national registry, a registry of patients that will record data about their experience on the drug that will allow us to learn more about the drug's risks and benefits. I'm hopeful that that might be part of an FDA process called a REMS, a risk evaluation mitigation strategy. Maybe it'll be part of a CMS process, but the more it's a national registry with national data, the more we can learn and move forward from this very exciting advance in our field. Dave, any closing remarks otherwise?
No, I think I'll just say, as Jason said, that our role is to help walk people through this process and through thinking about the drug and what makes the most sense for them as we move to hopefully a point where we'll be able to prescribe this medicine in the future. And so these certainly should be discussions that you have with your clinicians when you see them at your appointments here, and certainly something that I've already been talking to my patients about a fair bit.
Yep, doctor means teacher. That's what we're here to do. Back to you, Terry Casey.
All right. Well, thank you, Dave. Thank you, Jason. I appreciate it. And thank you to all of our viewers and attendees for joining us on today's conversation. We did accept a lot of questions ahead of the event that we then were able to work into the presentation. But if you have any questions that were not answered today, you'll be able to visit the Lecanemab Leqembi FAQ that will be posted on pennmemorycenter.org. You all can also continue to email either to the general PMC address, which is pennmemorycenter@pennmedicine.upenn.edu, or to me directly, which is a little bit easier to remember, which is tjcasey@upenn.edu. And we will work all of those questions. If we can answer them, or if we can, we'll answer them directly to you. If they have a broader interest, we'll work it into the FAQ page, and that will be up around the same time as this video. So thank you again, Dave, and thank you, Jason, and we'll look forward to the next one.
Bye, everyone.